All. This is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. So, the discussion today is really important, and this is about the autophagy and when does this start in our body. I'll give you the summary right now in two sentences, and then we'll go into detail. There is the most latest study in vivo rat or mouse models that says 8 hour and 16 hour split creates sufficient autophagy to help us recover from many of the issues for which we are doing autophagy. I would go in detail about this. Previous studies have shown that autophagy actually begins at the fasting of 24 hours. This is also animal studies, not human studies. And it peaks at 48 hours. That is quite harsh for someone who is used to 2 or 3 meals and snacks every day. So do not attempt that without actually thinking, planning, talking with your doctor, considering your health status, and then doing it. So that is the summary of the whole discussion. Now I'm going to go into all the nuts and bolts of this discussion with the mechanisms involved. There are three mechanisms I want to discuss: macroautophagy, microautophagy, and chaperone mediated or chaperone assisted autophagy. They all have a different mechanism, and they all have a different way of behaving, and they all have a different milestone when they become active. So, you know, the main story: 16-hour, 8 hour is the best form of intermittent fasting for autophagy's reasons. If you can actually compress your meals even more than 8 hours, for example, 6 hours or 4 hours or one meal a day, that is the best form. But if two meals, then 8 hours apart, no snacks in the middle, and then 16-hour of the gap. Now, I'm also going to talk about sleep with that as well for neuronal autophagy. So let's do that. Everything cannot be packed into the uh, summary. So let's start a discussion. So this is drbeen.com. Many of these lectures end up on drbeen.com as well. And there are 900 more lectures over there too. So if you would like to take advantage of getting access to drbeen.com premium content, it is lesser than $100. It's a one-time fee. There is no recurrence. And I think you would love the lectures. There are immunology and bacteriology and cardiovascular system and respiratory. There's a lot of things over here. So take advantage of that. The link to this plan is in the description of this video. I'm going to close it because I have too many tabs open. This is Yoshinoro Ohsumi. He is the research worker who was given 2016 Nobel Prize in Physiology and Medicine for his discovery of the mechanism and the genes for autophagy. Autophagy's discussion started somewhere in 1953 and 60 something, and so there has been research since then. However, this researcher here, I keep forgetting his name, Yoshinori Ohsumi, his work was the first to actually show or demonstrate the genes and the mechanism of autophagy. That is the one from where the research on autophagy exploded after he found those mechanisms. So that is that. Then the latest study that I discussed, this is the Professor Dr. Anna Maria Cuervo. She is a professor of medicine. She is an MD as well, and she does research on autophagy and other molecular mechanisms. This is her latest research about 8 hours and 16 hours gap. We'll discuss more, but this link is present in the description as well. With this, I'm actually going to close a bunch of these links because there are just too many links here. However, you can have a quick look at the references that are present. These are in the description as well. So after leaving these few, I'm going to close the rest. As you can tell, I was studying the whole day today to present this to you. So I'm going to just close them. All right. So let's start with my drawing. Don't you sometimes find it crazy that I read and underline and then I draw and I just have so much fun with it? Okay. So why is autophagy important? So please remember this when we discuss in layman discussions that autophagy is triggered or activated or stopped. Autophagy can never be stopped. Our cells always need to perform autophagy. Similarly, autophagy activation simply mean going beyond the normal balanced mechanism that it is there and boosting the autophagy. Autophagy is never switched off and never switched on. If that happens, we'll die. So it is really increasing and reducing the pace of autophagy. So why do we need to do the autophagy? Look, every second, just look inside of your own self. Just close your eyes and think about yourself. Every second, 3 million new red blood cells are being formed every seconds. So this 5 second, 15 million red blood cells are formed in all of us. Every second, 10 raised to power 15. I don't even know how many billions and trillions that is. 10 raised to power 15 new molecules of hemoglobins are formed. And also remember that as these new proteins are formed, in an equal amount is degraded as well. We stay mostly in a particular structure and shape. Mostly. That means what we make versus what we break is usually in balance. If we make more and break less, we'll start gaining weight, which happens. Or if we break more and make less, then we would start losing weight. That can happen too. But normally, in a healthy person, there is a steady balance between them. However, there's a lot of amount of proteins being made. Then, for a 60 kilogram person, for example, they make every day 240 gram of new proteins or proteins. So every day we are making more red blood cells, we're making more white blood cells, we're making more antibodies, we're making more complement system, we're making more, let's say, D dimers, and we're making more platelets and so on. So we are making things all the time. Actually, do you know that every 3 months, every protein in our body has become replenished with a new protein? We become renewed every 3 months. And I had that discussion in the past as well, that even in our brain, for example, hippocampal area, that is related to the memory and limbic system, emotions, intelligence, processing new memories. Hippocampus has 700 new neurons formed every day. That means 700 neurons are recycled every day as well. I hope I'm making a point that it is important that we understand that autophagy occurs all the time. So there is no concept of stopping the autophagy or starting the autophagy. It's really changing the pace. Now, when we make 240 milligrams of proteins daily, normally from outside we gain or we get about 70 grams. And then the remaining 170 are actually recycled from inside. We break down existing proteins, we take their building blocks, amino acids, and we make new proteins. So if autophagy was not happening, then all of this will not work. Now, ubiquitin, today we'll discuss two mechanisms. There is a mechanism of recycling protein, which is called ubiquitin-based mechanism. That mechanism only occurs for very specialized targeted proteins, not for every protein. And the Nobel Prize for ubiquitin discovery or the mechanism discovery was in, I believe, 2014. Aaron and Avram and Irwin were the Nobel Prize receivers in chemistry. Then in 2016, as I said before, Yoshinori Ohsumi of Japan. He received a Nobel Prize. I saw his name written. I believe this is Japanese, but I just wanted to write it as well. So that is in his honor. I wrote his name in these. I have no idea what that says. I'm sure his name. Okay. So now, autophagy, when does it start? So far, the studies have multiple ways. How am I going to explain this? Number one, in terms of hours. Number two, in terms of our body's hormonal statuses. And number three, in terms of therapeutics. Number four, in terms of our body habitus. So there are multiple ways to look at autophagy start and stop, or actually acceleration or not. So first, begins in 24 hours. This is the most common studies. Begins in 24 hours. So whenever you read somewhere where they say that short-term fasting starts autophagy, what they mean by short-term is 24 hour. That is really sure short-term, but we are usually not very used to such fasting. So be careful. 24 hours it begins. And how do we know it begins? The phagosomes, lysosomes, or autophagosomes or autolysosomes start forming. We have done that discussion in the past. Then it peaks at 48 hours, and then it sustains till a person dies or they would maintain their food. A person's health, their nutritional status, stress levels, body habitus, all that matters. For example, whenever a person is in ketosis, their autophagy levels are higher than those who are not in ketosis. Similarly, if somebody has a low glucose level, then they would have autophagy started. If somebody has low insulin levels, they would have autophagy started. Then if somebody is taking, for example, resveratrol, or somebody is taking, let's say, what are the other, coffee, there can be many factors that can be changing the pace of autophagy. In more recent studies, Dr. Anna Maria Cuervo, she is a professor at the Department of Development and Molecular Biology, and she's a professor of Department of Medicine as well in Albert Einstein College of Medicine. I was reading her work and listening to her talks, and she said that what they have observed is that macroautophagy starts within 8 hours of fasting. And this is important, that between the 8 hours, there is nothing eaten, no snacks. Fluids are fine, but meals are too, and these are strictly 8 hours apart. And then 16 hours after. In the first 8 hours gap, macroautophagy starts. And then in the 16 hours gap, the chaperone mediated autophagy starts, which is an excellent and intense autophagy mechanism. Very valuable mechanism. And I have done this discussion separately, that what is the benefit of autophagy? It is such a marvelous mechanism for healing our body and getting rid of the waste and trash and garbage that is stuck in our cells. Now, it is important to consider. So, if you didn't take away anything from this discussion, just remember 8 hour and 16 hours plus sleep in 16 hours. This is it. You can write it down somewhere. This is the whole thing to practice here. Sleep within these 16 hours of the fasting. Not at the boundary of 8 to 16, not in the 8 hours, but 16. Why? During the 16 hours, which is a prolonged fast compared to 8 hours, the microautophagy starts, and the neurons only perform autophagy when we are sleeping. So you could be hungry for 20 hours and not sleeping, and your neurons will not start autophagy until they know that you are sleeping. Because neurons are always working, and when you are awake, even if you're not doing anything. If you're awake, your neurons are firing and working and keeping your muscle tone and operating your body. They have a lot of things to do. They will not trigger autophagy at all. This is equal to if you're driving a car. Let's say you are the one who are driving the car, and while driving you leave all the driving, the steering wheel and everything, and you start dusting around in the car. That car is going to go in a ditch. So the neurons know that when we are functioning, we are not going to do any cleanup, we are not going to do any recycling. So the 16 hours time for your fast should include the sleep time. That is the time when maximum intense autophagy is occurring. And if you allow your brain to rest at that time, that would help replenish, recycle, and clean up the neurons as well. If you get this much, that's it. We don't need to listen to anything else from Dr. Mobeen. This is enough of a valuable asset even for the rest of our lives. This is such an important thing. Okay, so I think this message is done. Let's go to other ways of understanding when autophagy would start. For example, when glucose levels go so low that glycogen is also used up from the liver. So what happens is when we are fasting, and I had promised that I would do this discussion of when do we use various nutritional stores. I actually asked the patrons today to vote on it, and they voted on this topic and not on that one. So what happens is when we start fasting is usually 2 hours after a meal. For 2 hours after a meal, we continue to use the food or nutrients from the meal. After 2 hours, we have either used up the nutrition or we have stored it up. And now we are hungry. Now we are fasting, or we shouldn't call it starving, but we are hungry. Now we're going to start pulling from the stores and start using it. So you can say that what kind of a body is this, that you just finished storing and you turn around and say, all right, open up the stores, I want to use them. But that's just how it works. So when you start using the stores, the first stores that are depleted are going to be the glycogen in the liver. Glycogen in the liver is the stored form of glucose. Usually, it takes 14 to 16 hours for the glycogen levels to go drastically lower, that our body says, I don't have much more glycogens available, I need to do autophagy. So 14 to 16 hours glucose levels. So that is another milestone. Glycogen levels low. Now, attached to that is also the caloric restriction. You could do 8-hour meal separated, but you could also restrict calories. And that would also accelerate and intensify autophagy. Then you could decide what kind of meal you're taking. Fat heavy meals or 70% fat, or we all know ketogenic meals can trigger autophagy as well. Actually, anybody who is in a ketosis has autophagy accelerated as well. So that's a milestone as well. So you don't have to wait for some time. You can say, all right, I'm going to go into the ketone state using a ketogenic state, nutritional status or plan. And as soon as I start having ketones, I know I have autophagy. Or you could look at your blood glucose and insulin levels, and as they drop, you would start entering the autophagy. Now, if you ask me, when do they drop? Within 2 hours, we go back towards normal. And then we can sustain that normal by breaking down glycogen and glucose stores that are in the stores and free up the glucose for some more hours. And then 6-7 hours later, we actually don't have that as well. And now we need to get it from autophagy and from other stores. Now, there is an interesting study that showed that if you put human cells, so there are very few human studies on autophagy. The reason is that you cannot just ask a person to starve and then say, I'm going to take some cells from here and some cells from there. So less studies. So what they did was they took human cells, put them in a petri dish in vitro, and they made them hungry. And they found out that within 24 hours, the neutrophils started performing autophagy. This would actually not translate equally to our body because the neutrophils sitting in a petri dish are not really performing anything, they're just sitting there. The neutrophils that are sitting in our body are actually working all the time, so their autophagy should actually start earlier than 24 hours. But this is the study. At the same time, please remember, if somebody is pregnant, so the child needs nutrition, their child needs proteins, they need proteins, they are contributing a lot to the child. Similarly, breastfeeding mothers, athletes, hyper active um, activities, soldiers. So there are many times when triggering autophagy will not be correct. So one has to be very, very careful. Cancer patients, now cancer patients and autophagy is a delicate matter, because what happens is, on one hand, we want autophagy to occur so that the newer cells that are bad cells, they die. On the other hand, when the autophagy occurs in cancer patients, sometimes the immune system cell dies, but the cancer cells continue to move forward. And that actually gives the cancer cell even more of a boost because the attacking cells are gone. So it is very important for cancer patients to figure out what is the right balance of autophagy. Similarly, folks with the immune issues, for example, if they have hyperimmune issues, then autophagy is actually useful by clearing their cells and reducing their cells or reducing the activity of the cell. On the other hand, if they have a reduced immune response, and then if we trigger autophagy or starvation of their cells as well, then their immune response can become even lesser. Now, what are the signs of autophagy? Some signs. Look, when you can detect ketones, there are urine tests, right? When somebody is in ketosis, they have autophagy accelerated. When somebody's appetite reduces after hunger, they were not eating, they became hungry, they started starving, and then suddenly they became calm. The appetite went away. That is because the autophagy has started. Autophagy, because of some unknown mechanism, suppresses appetite. So after the hunger, loss of appetite could be a sign of autophagy. I mean, loss of appetite can be for pathological reasons too. Then fatigue, development of fatigue. Now remember, just like in ketosis as well, or ketogenic diets as well, there is a brick wall that we hit and then we become okay again. Similarly here, when the autophagy starts, we have a temporary wall of fatigue. And that is because the calories are less, glucose levels are less, stress hormones are going up because body is under stress. The cells are saying, what the heck, we don't have food. They are sending signals. So the body is in that state, after which the body is going to say, you know what, I think this is what our life is. We are starving. And let's start our autophagy and start doing the recycling. Bad breath is in ketosis. The acetone present during the ketosis causes bad breath. That doesn't mean every bad breath is sign of autophagy. Not you running around, hey, look, I have bad breath because I have autophagy. When you are in ketosis and the bad breath can be smelled, acetone breath, that is also a sign that ketosis, this autophagy has accelerated. Weight loss, for sure. So when people are losing weight, they are starving their system, and that is causing accelerated autophagy as well. And if you actually notice improved brain function, and it is interesting that we fast, and that fast is quite a rigorous fast from the sun, uh, before the sunrise to after the sunset. Sometimes these fasts are very long and there is no food and there's no water and nothing. And one becomes very sharp during that time. So, this is one of the signs of autophagy, that brain functions become sharper and focused and you become more active. Then here is another study, intermittent fasting and obesity related health outcomes. So in this study, they said outcomes associated with modified alternate day fasting. So this is alternate day fasting, 1 day fasting and 1 day eating. Alternate day fasting included a moderate reduction of body weight, body mass index, and cardiometabolic risk factors in adults with overweight or obesity. So this is another way of causing autophagy and having the benefits of it. Again, a little more rigorous, to not eat for 24 hours and then eat for 24 hours. And I think Dr. Paul Merrick has mentioned it publicly, so I can probably share it as well. Excuse me. He's doing intermittent fasting and he eats just one meal. And he said, I have weight loss, I become more active, I'm more active, I feel more healthy, and so on. I think he feels more active as well. So 16-8, sleep during the 16. Now, last part of this talk today, types of autophagy. Macroautophagy, microautophagy, and chaperone assisted or chaperone mediated autophagy. These are the three I'm going to explain and then we break. So, types of autophagy. Macroautophagy. Here is what happens. So, here is a cell. In the cell, imagine there is some trash in the cytoplasm. I have not made the nucleus. Somewhere here is a nucleus as well. So, this is the cytoplasm, and inside the cytoplasm, there is some trash. There is some bad, unwanted, unnecessary proteins hanging out or hanging around. What happens is within the cell, a structure starts forming called phagophore. And this is controlled by ATG genes, autophagy genes. And then there are autophagy proteins that are formed when those genes are expressed. And that these proteins are the ones that do these actions or that help this action. This is what Nobel Prize was given in 2016. So when we have hit the autophagy reason, whatever that reason is, it's the starvation or the therapeutics or other things. When we have hit that, a structure starts forming, a tiny ball starts appearing, which is called a phagophore. And that phagophore starts becoming filled. It's like a little Pac-Man that start crunching, if that is a word, start eating away pieces from the cytoplasm, the trash. Imagine a tiny little monster that is just eating trash. So it starts forming with the open mouth and eats trash and closes mouths. When it closes mouth, it becomes a phagosome. Now, the endosomes for the medical professionals and students, endosomes are when we are capturing things from outside and bringing them in, right? Phagosomes or endosomes. Even those endosomes can also come in and become fused with the internal phagosome and make a bigger particle, meaning these two pathways can combine together. At the end of the day, just visualize that there is some trash inside the cell, and there is a little monster Roomba that comes in and picks up that trash. Once that trash is picked up, it's a double phospholipid bilayer. And then it fuses. And why did I stress it is a phospholipid bilayer? Because it's a double layer. Normally inside the cell, the vesicles are single layered. However, from outside the cell, when the vesicle come in, they are double layered. And if we make autophagosomes, they are double layered as well. And the importance is we can detect them as double layers. And we know phagocytosis or autophagy is going on. So anyways, here is a phagosome that is a double layered vesicle. It fuses with the lysosome. Lysosomes are little stomachs of the cell. They have a lot of acid in them, and they have a lot of enzymes that can break other structures like proteins or DNAs or RNAs and so on. So now we fuse them together. This fused set is called autolysosome, and these acids and the enzymes, they will now pour into this little trash bag, and they would digest the trash in the trash bag. This is called macroautophagy. So if you are eating meals at 8 hours gap without snacks, you are setting up your body for the macroautophagy. It is important. Then, in these 8 hours, microautophagy can occur as well. What is microautophagy? In microautophagy, instead of forming a little blob in which we fill the trash, instead of making a trash bag with the trash in it and then bringing it to the shredder, the shredders, look at this little cute red thing. It directly goes and attaches to some part of the cell and just munches it away and takes a bite out of it and breaks down that bitten part. So here the lytic vacuole, the lytic vesicle, the lysosome directly attaches to the protein to destroy, and it directly munches it away. It does not need this intermediate phagosome. When I was drawing this, I was visualizing as if there are little monsters that are biting into the walls and just munching away, little rats, let's call them. So these are those little things. They can break down DNA pieces like this, or broken pieces of DNA, or damaged parts of DNA. They can break down endoplasmic reticulum. They can break down parts of Golgi apparatus. They can break down a lot of trash in the cell. Macroautophagy, microautophagy, these both are within 8 hours gap, and they both become primed and active during that time. Then comes the 16 hours, more long one. During this time, the heat shock proteins become active. What are heat shock proteins? Heat shock proteins are within our cells. We have sensors, we have proteins whose job is to figure out if the cell is under stress. How can a cell be under stress? A cell can be under stress if you're exercising. What are you doing when you're exercising? If you look at my muscles, the muscles are moving and they're changing their shapes and they're becoming D-shaped, and that cytoskeletal change. Every cell has a skeleton. Just like we have a bigger skeleton, our cells have tiny skeletons as well. And when that skeleton stretches and skews and changes shapes, that causes the heat shock proteins to become active. So just by exercising, you can induce autophagy. If you didn't do anything, no starvations, no hungers, nothing, no intermittent fasting, no resveratrols, and no coffees. If you just exercised, you would trigger autophagy. So when the body is under stress, or body is starving for 16 hours, 14 hours, 16 hours, glucose levels are low and nutrition is not there, even then the heat shock proteins become active. They are called heat shock because they were first observed during heat shocks of a cell, but now they're found out to be activated during any stress, including stress of exercise. So when they become active, there are many kinds of heat shock proteins and they have various functions. One of the function is to act as a chaperone. What they do is they go around in the cell and they find out trash material. They know the cell is under stress. They know there is going to be junk and trash present now. And they also know the cell is going to need more nutrition and it is going to need more energy. So they go about figuring out broken material, wasted material, trash material, unnecessary things lying around, and they would pack them up into small structures. These are called, over here I've made that as a trash can, but these are called substrate. And this is a chaperone. So chaperone or ubiquitin becomes actually not ubiquitin, heat shock protein becomes connected with the protein to break down. And then it brings it directly to the lysosome. Remember, the lysosomes are those little pockets of the acids, the fireballs of the cell, right? So these heat shock proteins would chaperone these trash waste bins to filled with the trash material to this vesicle. Now, here is a wonderful thing. Well, prize went for some reason. The vesicles have a specialized protein called LAMP2A. This LAMP2A specially allows the trash brought in by the chaperone to move through the membrane and in the stomach. This does not happen otherwise. This only happens with this chaperone mediated system. That you could directly spill the trash in this stomach vesicle, lysosome, if it is a heat shock protein that brought it over. Not everything else can do it. Not every protein can cross this membrane and enter the vesicle. Only those proteins are given special protocol with a special gate to enter, and that gate is LAMP2A, and the chaperones are heat shock proteins. This is CMA, chaperone mediated autophagy. This happens after 14, 16 hours of starvation. This causes the best cleaning up of the cells. And this is what is the part that would come after 8 hours for the next 16 hours. This is the time when you should be sleeping as well, so neurons get cleaned up as well. Please let chaperone mediated autophagy to work. Whenever you're feeling less healthy, whenever you're feeling depressed, not able to memorize, having cognitive declines, having fatigues, or feeling allergies, give it a try. Of course, talk with your doctor, figure out what is right, how much of a fasting, but give this try. CMA is the best form of autophagy. And then, last part. This autophagy, M. Gregory said that donkey is so cute. Imagine when I was drawing these things, I was just cracking up and I was drawing. Now, last slide. This is a regular mechanism of recycling proteins. Our cells actually have two main mechanisms for recycling. One mechanism is called ubiquitin-based mechanism. Other one is called lytic mechanism. The mechanisms that we just saw, the autophagy, these are all lytic mechanisms. Macroautophagy, microautophagy, chaperone-mediated autophagy or chaperone-assisted autophagy, these are all called lytic mechanisms because there is lysis, there is digestion by lysis in the stomach, cell stomach. This mechanism is called ubiquitin mechanism. This is a separate pathway. It also does the protein recycling. However, this pathway is very selective. It doesn't willy-nilly pick up any piece of trash and fixes it. It will only decide which protein to work with. That is as if there is let's say there is a party, and after the party, there are a bunch of paper plates and cups and stuff sitting at the table. And somebody comes and says, I'm going to help you with the cleaning up this trash. And they pick up their own paper plate and go and put that in the trash bag. And that's about it. And they said, thank you very much. I helped. But somebody else could come in and pick up everything and bring it to the trash bag. So those who picked up everything without regarding from where this trash come from, that is lytic mechanism. That is autophagy. However, that system that is very targeted and selective for which protein they would break down is ubiquitin mechanism. That is why ubiquitin is not that helpful as autophagy. So in the ubiquitin, what happens is, imagine this is this little purple protein needs to be broken up. So this is a ubiquitin protein. You mark the protein, you tag the protein with ubiquitin, and then those proteins with their tags are brought to another vesicle in the cell called um, proteasome, endosome as well. Proteasome's job is to break proteins. So these little proteins are brought in and thrown in these proteasomes. Proteasomes would shred them and break them apart and put that raw material out. Then that raw material will be used to make more proteins. This is ubiquitin mechanism. Ubiquitin mechanism is separately always working. Autophagy is separately always working. We are just figuring out now how to use autophagy for benefiting and healing, or trying to heal, chronic diseases, chronic fatigues, neurological issues, immunological issues, liver diseases. We are trying to figure out how to modulate it in cancers. Now, the spike protein issues, we are trying to figure out how to use autophagy with the spikes as well. And dare I say that I think FLCCC and my talks there are the leading front for autophagy in the spike proteins and long COVID and vaccine injury. So this is the discussion. I hope you liked it. Um, there was a bunch of love went in it to draw them, but I enjoyed it as well. So please do me a favor. Please like, subscribe, and share. The smallest fee you can give me is to like. And if you would like to support this work, actually, I wanted to thank everyone. You have kept me independent of having other people as my boss. You are my boss. And you kept me independent by supporting my work. So if you would like to support this work, in the description of this video, there is a link to drbeen.com. You can get access to 900 more videos other than these for a very reasonable price, actually illegally reasonable. Then there is a link to use PayPal if you would like to use PayPal to support this work. There's a link to use to buy me a coffee if you would like that, or you can become a patron. I like it that patron, $5 per month, and they can just join and be there, and we sometimes discuss things and vote. So this is what we have. Thank you very much for your help, for your support. And just like uh, Siddhartha, thank you very much. You can become part of YouTube as well. So thank you. I hope you liked it. Now for the chit chat, I am actually doing one more lecture for which I have to do a couple of drawings. Would you like me to join the chit chat on the cafe side? And I will be drawing. Maybe we can discuss while I'm drawing as well. Maybe you can draw with me as well. Or would that be too much of a distraction? Okay, so John says yes. So we can go to Cool Bean Cafe. I have to do the other lecture, so I have to do some drawings. We can do them together, and then I'll do the lecture after I hang up. Okay, so I'm going to see you there. I'll share the link with Margaret. I'll see you there. So once again, please like, subscribe, and share. I'll see you soon. Bye-bye for now.